Hi. Okay, this week we're going to go over the parathyroid gland and how it plays a role in calcium and phosphate regulation. More of this is going to focus on calcium than phosphate itself. So let's get started. The regulation of plasma calcium levels is essential for normal cell function, neural transmission, membrane stability, bone structure, blood coagulation, as well as intracellular signaling. Now, the balance of plasma calcium levels is really dependent on three components. Parathyroid hormone, henceforth referred to as PTH. This is a hormone that is made in the parathyroid glands. Vitamin D3, that's the active form. We get that mostly from dietary. And calcitonin, which is made, interestingly enough, in the C cells in the thyroid gland. Now, PTH stimulates bone resorption and the release of calcium into circulation. When I talk about calcium, I'm going to be talking about the calcium ions. They have, of course, a +2, but to shorten things, I basically just refer to as calcium ions. Now, in the kidney, PTH promotes calcium reabsorption and inorganic excretion of phosphorus in the urine. PTH stimulates hydroxylation of 25-hydroxy vitamin D. Now, this is a hydroxylation that occurs at the 1 position. This results in the formation of 1,25 hydroxy 2 vitamin D, which is the active form of vitamin D. It's also called calcitriol. Now, vitamin D increases intestinal absorption of dietary calcium and facilitates renal absorption of filtered calcium ions. Now, in the bone, vitamin D increases bone reabsorption with a resulting increase in the release of calcium ions into the blood plasma. Calcitonin counteracts these effects of PTH by inhibiting bone reabsorption and by increasing renal calcium ion excretion. The overall result is that PTH, vitamin D, and calcitonin maintain normal plasma ion, calcium ion concentrations. And you can see this in a diagram in your text, okay? Basically, as you can see here, when we talk about the kidney, we're talking about from the filtrate, we increase the amount of calcium that is brought back in, so it maintains itself in blood plasma. Now, when we have cells that detect calcium levels, they are going to basically control the amount of PTH released, or if PTH release needs to be decreased. Notice also that calcium ions will begin to be liberated because of the bone resorption, in other words, converting the calcium phosphate, otherwise known as hydroxyapatite, into calcium ions and phosphate ions. Okay? So really PTH's targets are going to be the bone and the kidney. Okay? With the kidney, you also are going to have an increase in vitamin D activation, again caused by PTH. And you will also have an increase in calcium ion reabsorption. And you have the phosphate. Now, phosphate, PI refers to phosphate that is not attached to any carbon molecule. So hence it's inorganic. And this is going to be declining as a reabsorption. So it's going to show up more in the urine. Now, let's talk about the functional anatomy of the parathyroid gland. Parathyroid glands are really these kind of P-shaped glands located at the top and bottom of the posterior borders of the lateral lobes of the thyroid gland. The cells that secrete PTH are called chief cells. Now, there's another set of cells there called deoxyphil cells, and the function is really not known. These exist also in the parathyroid gland. Excuse me. Now, the PTH polypeptide hormone is about 84 amino acids in length, and it has a continuous release, although the book mentions periodic bursts that will increase its release. And this is usually in response to things like declines in the amount of blood plasma calcium levels. The central role of PTH is involved with bone remodeling, as well as calcium ion homeostasis. PTH hormone is degraded, and basically the time period is, it's degraded by the liver, and its half-life is about 5 minutes. The regulation of PTH release. This is a very tight negative feedback system. And it's based on calcium plasma levels. High calcium levels are going to suppress PTH release. Low calcium levels are going to cause a high release of PTH with a reduced PTH degradation. And here you can see a diagram of the parathyroid. The gland itself, they're really, really small. And as a matter of fact, in situations where doctors really didn't understand the importance of the parathyroid gland, they would do a thyroidectomy and end up removing the parathyroid glands as well. And this led to all sorts of calcium ion level irregularities. As you can see here, the cells, the chief cells, are distributed excessively in those glands, and there are some of these enlarged cells called the oxyphil cells. As I've said before, they have no real known functions. Now, when we talk about the regulation of blood calcium, here you notice on this side you have high blood calcium levels, and so from the thyroid there are cells called C cells. They stand for clear. Okay, so clear cells. I'll show you that later on. They're going to release calcitonin, and that's going to provide for a buildup of bone by osteoblasts. Now, if you get osteoblasts and osteoclasts mixed up, think about this. Osteoblasts build. The C classes are osteoclasts are basically for the breakdown of bone matter. So when calcitonin gets involved, the blood calcium levels will decline. And it's a seesawing effect just to maintain the normal blood calcium levels. Now, moving on, what you have are the low blood calcium level. What you have here are the parathyroids, and they're going to release parathyroid hormone. Now, this is going to lead to a breakdown of bone material by osteoclasts, and the blood calcium levels will increase as a result. So let's talk about the PTH calcium sensing receptor. The calcium sensing receptor is a G protein coupled receptor. There's several different types. There's G alpha Q/11 and G alpha I. These are located in the PTH chief cell plasma membrane. These receptors are also present in the kidney tubule, interestingly enough, as well as the thyroid C cells. Release of PTH is inhibited by calcium binding to the sensor receptor. So basically, as your calcium plasma levels exist, some of the calcium will bind to the sensor, and that will start triggering a inhibitory reaction that will reduce PTH release. Now, of course, activation of the calcium receptor will trigger a signaling cascade. This involves the phospholipases of C, D, and A2. Of course, by activating these phospholipases, you're going to get a resultant release of the secondary messengers IP3 and DAG. They will be generated, and also you're going to have an arachidonic acid cascade. Now, of course, what happens in these cells is that by activating all of these systems, you're going to have an inhibition of PTH. PTH is going to be existing already in granules, ready for subsequent release. When you do the inhibition, this is going to lead to the degradation of the PTH-containing granules right off the bat. Now, it's interesting to note that during hypocalcemia, the calcium sensor receptor is relaxed, and the PTH is immediately being released. And you can see that here in this diagram. Here, calcium is attached to this sensor. And what's going to happen is a decrease in cyclic AMP and a decrease in PTH release. But if you have something like hypocalcemia, where there's no calcium ions attaching, you're going to have an increase in cyclic AMP and thereby an increase in PTH release. By the way, excuse me, you will have a really extensive inhibition on many of the cells if you have hypercalcemia. Now, let's talk about the regulation of parathyroid hormone release. PTH release is increased by hypocalcemia. That kind of makes sense. But also hyperphosphatemia and catecholamines. Now, the interesting thing is the PTH release is suppressed by hypercalcemia, which we kind of had expected. Also in significant amounts of vitamin D, as well as severe hypomagnesemia. Now, plasma magnesium in its ionic levels can also regulate PTH secretion. Moderate declines in magnesium ion plasma levels can trigger PTH release. Magnesium can bind to calcium sensor receptor. Also, low magnesium plasma levels usually reflect calcium levels in a similar way most of the time. So if you've got hypomagnesemia, you probably are also going to have hypocalcemia. Okay? But that's most of the time. Severe hypomagnesemia can inhibit PTH secretion, inhibit bone responsiveness to PTH-mediated bone resorption. And that's sort of the exclusionary principle here. Because I know students right now are probably going, well, severe hypomagnesemia, and yet you're getting suppression, and hypocalcemia, you're getting an increase. Right. Now, let's talk about the PTH target organs. Primarily what we're talking about is the bone and the kidney. The kidney we really deal with in the sense of excretion. The bone we consider as this calcium storehouse. The main physiological response from PTH to increase plasma calcium levels is by increasing calcium renal absorption, calcium mobilization from the bone, and of course we can increase our intestinal calcium absorption. But there's another player that's necessary there, and that is vitamin D. PTH also increases 1-alpha hydroxylase activity, and this you're going to see in the kidneys, as well as renal phosphate activity. The activity requires PTH binding to PTH receptors on the target tissues, of course. Now, here we have another chart that helps clarify some of these little points here. One thing you want to keep in mind is this is another endocrine system that is not under control of the hypothalamus nor of the anterior pituitary. We mentioned a couple before. We're going to go explore those in the subsequent weeks. All that this system is controlled by is the plasma calcium levels, and to a lesser extent, magnesium. And you can see this by a negative feedback mechanism. Plasma calcium goes up, PTH secretion goes down. Okay? And you can see this plasma calcium levels go down. You're going to have a release of parathyroid hormone. Okay? Plasma calcium goes up. You go here. And at the same time, what you're going to notice is that at a certain point, the thyroid C cells will be releasing increasingly the calcitonin. And then that's going to bring the plasma calcium, ionic calcium levels in the blood back down some. And here is another example of the negative feedback loop. We're going to consider just PTH for a second. PTH will act to stimulate intestinal absorption of more calcium. It will stimulate the bones to release calcium ions, and it will also stimulate the reabsorption of calcium in the urine. Now, all of these rising levels of calcium are going to feed back negatively on PTH release. And you can see how this is in this other diagram here. All of the increased serum calcium will negatively feed back onto the parathyroid hormones, reducing PTH secretion. Okay? When you have low levels of calcium, that is going to cause the release of PTH. Will work on the bone to release calcium, will work on the kidneys to increase calcium reabsorption, and it will activate vitamin D, and this will play a role in increasing calcium absorption from food. Now, let's talk about the PTH receptors. There are three types of receptors that have been identified: PTHR1, PTHR2, and PTHR3. All of them are G protein coupled receptors. Physiological function, though, for PTHR2 and PTHR3 is not known. PTHR1 is a G protein coupled using G alpha S subunit, and this leads to the activation of adenylate cyclase, an increased production of cyclic AMP, the activation of protein kinase A, and phosphorylation of target proteins that have serine residues. Now, the cellular effect of PTH. In the kidney, PTH stimulates calcium ion reabsorption. It also enhances phosphate excretion. It enhances the activity of 1-alpha hydroxylase. This is the enzyme, or these are the enzymes responsible for the formation of active vitamin D. So if you notice, what we're talking about is really 1-2 punches or 3 punches to bolster up quickly the calcium levels. Now, the site of the PTH regulation is the renal distal tubules. PTH regulates active transcellular calcium pumps. In the distal tubules, calcium absorption is transcellular, crosses the cells. It's regulated by vitamin D, PTH, and calcitonin. PTH decreases renal as well as intestinal reabsorption of phosphate by decreasing the expression of type 2 sodium phosphate cotransporters. And you can see this diagram that's in your text. You have your interstitial space here, your distal tubule here. Calcium is coming through. It is positively enhanced for the calcium channels, ion channels, to bring in that calcium. It'll be held for a bit by calbindin D, 28K. Vitamin D will play a role in both the presence of that calbindin, but also in the calcium ATPase, which is basically a pump. And what is it going to do? It's going to take energy, ATP, and hydrolyze it till you get ADP with PI, and it's going to pump out the calcium. Now, as we're pumping out this, you can also have the calcium sodium exchanger. For every 3 sodiums, you have a calcium coming out. That's a basolateral at the basolateral membrane. You can see this. It's a little fuzzy, but it's a similar photo. And what is going on is that as PTH is working out here, this is more sort of a phosphate attempt at balancing things out. You have the tubular lumen here, and you have the basolateral membrane here. And what happens is lysosomal destruction of that sodium phosphate transporter. Here's the phosphate, here's the sodium. PTH revs up the breakdown of these pumps. So you allow more of the phosphate to be lost. All right. You do have some phosphate come in. This is the sodium phosphate grouping here. And you also have a means to balance out the sodiums and the potassiums by the sodium-potassium ATPase pump. Okay? Cellular effect of PTH. In bone, PTH releases calcium and phosphate via increasing bone resorption. It also plays a key role in bone remodeling. PTH binds to receptors on osteoblasts, causing the cascade of cellular events resulting in the activation of osteoclasts. Osteoclasts are the key, demo crew for solid bone. Osteoblasts will activate the osteoclast by this PTH-stimulated event of Rankel L. This is a receptor activator of nuclear factor Kb, and this is a ligand, so they call it L. Now, rank is expressed by hemopoietic osteoclast precursor cells. So the activation via binding from rank to Rankel results in the maturation of osteoclast cells. So although osteoblasts play a little bit of a role, the worker that's going to actually do all the destruction of the bone and allow for this resorption to occur is the osteoclast. Osteoblast activation will result in several other activation of genes essential for bone degradation. Collagenase 3, growth factors such as insulin-like growth factor. Collagenase 3, just to help you to remember, bone is not merely calcium phosphate in the hydroxyapatite form. You have the reinforcement of the structure of bone by these fibers, collagen, and you have to break them down as well as the mineral. Now in the bone, the osteoclasts are going to produce acidification of the extracellular microenvironment via hydrogen ion ATPase, and that's going to be recruited from the osteoclast's ruffled membrane. In essence, what they're going to do is start pumping out and dropping the pH in the localized area. When you pump down to about 4.0, that's going to lead to the dissolving of calcium phosphate. Okay. Osteoclasts will also use transcytosis for calcium ion delivery to the systemic circulation. They have to pass it literally through their cells from endocytosis to exocytosis. And you can see a little bit of this demonstrated in this diagram here. And you can see how this works on the bone remodeling. Okay? This is the demonstration of PTH playing a role in osteoblasts causing the rank L. And then what happens is the precursors of osteoblasts will have rank, they will have binding, and that will trigger the maturation of osteoclasts. Keep in mind also that as transcytosis is occurring, you're going to have calcium ions released into the circulation. Okay? The osteoclasts are going to basically have released lysosomes. They're going to recruit hydrogen ions so that the pH is appropriately dropped lower. You're going to have the solubility of calcium hydroxyapatite, and you're going to have the acid proteases break down the collagen. Now, when we talk about calcium homeostasis, we have to think about this from the perspective of what is all this around? Why do we have this? What's necessary? Where does it work? And where is it stored in our body? How does it get transported? Well, let's start off. Human body has approximately 1,100 grams of calcium. 99% of it is deposited in teeth and bone. Calcium found in the plasma, though, 50% is in the ionized format. 40% is in a protein-bound structure. The majority of that protein will be albumin. Finally, we get calcium that's complexed to citrate and phosphate, forming soluble complexes. That's about 10%. Okay, that's 100%. Now, during acidosis, though, it's interesting. As the pH is dropping in the blood, protein-bound calcium decreases and ionized free calcium increases. During the opposite, during alkalosis, going above the 7.45 for the pH, you're going to notice that there's an increased amount of calcium ions that's bound to the various proteins, and the ionized free calcium will decrease. And you can see this here. Here's our calcium in plasma, 9.5 milligrams per deciliter. 40% is bound to protein. 80% of that 40% is albumin, 20% is globulin. Now, the free ionized, biologically active amount is 50%, and that's regulated by both PTH and vitamin D. This is the actual formula for active vitamin D. Now, here's another chart to help you to review this. When we talk about the interaction of bone, kidney, and intestine maintaining calcium homeostasis, we have to consider the following points. For the normal plasma concentrations of calcium ions, the range is between 8.5 to 10.5 milligrams per deciliter. This is going to be regulated via vitamin D, PTH, and calcitonin. The bone is a readily exchangeable pool, as well as being a very stable pool of calcium. The kidney, virtually all filtered calcium ions are going to be reabsorbed. Most filtered are in the proximal tubules under a passive process. So you've already got at the proximal tubes, the tubes that are closest to the glomeruli, a passive process that's going to pull in a lot of that calcium. But another approximately 40% is absorbed under hormonal regulation. And you want to keep in mind one other thing. After the growth phase, there is no net intake or loss. The amount of calcium loss in urine is equal to the net calcium absorption. And that's after growth. Okay? Now, with the intestine, we have a dietary intake, roughly about 1,000 milligrams a day. Yet only 30% is absorbed by the intestinal tract. The dietary intake is absorbed is enhanced by vitamin D during growth, pregnancy, and lactation. So under these three constraints, you're going to have a greater absorption of calcium in the intestine. And you can see this in another diagram, which actually also still exists in the Molina text. And you can see how you have 50% daily intake absorbed in this case. You're going to have some constant intestinal loss. But you're going to have minimum urinary loss unless you have some imbalance between PTH and calcitonin. The players here would be more PTH and vitamin D. Here, keep in mind that the players for bone is mostly going to be PTH and calcitonin also. And what I mean by that is they're going to have this influence of how this is basically sequestered or resorbed. Now, let's step away for a minute and go over vitamin D and calcium homeostasis. Vitamin D is a lipid-soluble vitamin synthesized from plant-derived precursors or through the action of sunlight from cholesterol-derived precursors found in the skin or obtained from dietary sources such as fortified milk, fatty fish, cod liver oil, or eggs. And by the way, fortified milk. You see this in all your milk containers, fortified with vitamin D. Why? To enhance the absorption of the calcium that is present in the milk, especially for growing young bodies. Now, active vitamin D is also called calcitriol and requires 2 consecutive hydroxylation steps, first in the liver, then in the kidney. And it has to be done for the precursors. Cholecalciferol, vitamin D3, is derived from the skin. And ergocalciferol, and that's the vitamin D2, is derived from the diet. When you finally get active vitamin D, which is the 1,25-OH2D, it's known as calcitriol, which circulates and is bound to the vitamin D binding proteins. PTH stimulates the activity of renal 1-alpha hydroxylase. This is going to increase the active form of vitamin D. This enzyme right here is key to making really an active version of vitamin D. Now, vitamin D and high calcium levels will suppress, interestingly enough, the activity of 1-alpha hydroxylase. You can see this in this diagram here, starting with 7-dehydrocholesterol, which is in the skin. And by the energy of ultraviolet light in the spectrum of UVB, you obtain a pre-vitamin D3. By heat, such as the body, you can convert this to vitamin D3, and you go through the first hydroxylation here in the liver, and now it becomes 25D. Now, by the way, we can't forget about the diet or supplementation, which is basically going through absorption, or in this case, it's going to go first into vitamin D3. Now, after it leaves the liver, it's 25D. You go to the next site, which is the kidney, and that's where you're going to have your second hydroxylation, okay? And you now have active vitamin D3. In the bile, you have inactive metabolites. They may be reabsorbed through the small intestine. But what you see here is this, that vitamin D that's active will act on the bone, immune cells, liver cells, etc. And also there is an enzyme that will inhibit and that will be involved with inactivation and excretion. Now, this is a little bit of detail I just want to throw out to you that you don't have to know the exact numbers, okay? But just see that, for example, dietary calcium, as it passes through the duodenum, small intestine, you will get some loss in the feces. You will have absorbed calcium. You will also have a secreted endogenous fecal calcium of about 150 milligrams, which contributes here because we did say that about 40% is absorbed, okay? So you end up with a net calcium absorption of 250 milligrams. You notice the bone here is going to be contributing, and it gets to the kidney, and the kidney is going to basically pass out 250 milligrams per day in urine of that calcium. And if you notice that you have these various situations of decrease in calcium intake, decrease in serum calcium, that's going to trigger the increase in PTH. Okay? And keep in mind that DT stands for distal tubule, so this is the increase in distal tubule calcium reabsorption. You also have an increase in the bone resorption, and you have the reestablishment of serum calcium. So the role of vitamin D. Do we have deficiencies? Yep. And deficiency of vitamin D due to decreased dietary intake of vitamin D, or lack of sunlight, or decreased conversion of inactive precursor in the skin to active forms of vitamin D can result in health problems. Now, lack of sunlight, just want to bring this up to you. If you look and Google it, you will find either in Siberia or they'll say it in Russia, where they don't have fortified milk. So what they do literally is get little kids to maintain their growth as they put on welding goggles, dress them down to their skivvies, and they are around a certain light that produces a tremendous amount of UV, so they make their own vitamin D. In this country, we don't do that. We basically fortify our food with vitamin D. Now, of course, vitamin D deficiency will result in bone deformities. In children, this is rickets, a softening of the bone, and you can see this. The vitamin D deficiency in adults would be a reduced bone mass that we call osteomalacia. Also, excessive intake in vitamin D can cause some problems. Calcinosis, this is a calcification of soft tissue. Deposition of calcium and phosphates in the kidneys. You know what that's talking about? Yeah, kidney stones. Also, an increased plasma calcium levels resulting in cardiac arrhythmia. Now, let's go into calcitonin. This is a 32 amino acid long peptide hormone produced by cells of the neural crest origin, referred to as the parafollicular or C cells in the thyroid gland. These are cells that are off the side. They are not related to the follicular cells that make T3, T4. Okay? The release of calcitonin is regulated by plasma calcium levels through calcium receptor on the parafollicular cells. Elevations of plasma calcium levels above 9 milligrams per deciliter will stimulate the release. Calcitonin's half-life, though, is only 5 minutes. It's metabolized by both the liver and the kidney. It is interesting to note that the release of calcitonin can also be stimulated by gastrin. Gastrin is one of those gastrointestinal hormones. We'll probably discuss at one of the last lectures, but it is more of a GI tract hormone than anything else. Here you see the clear cells. That's with the C cells. Don't get them mixed up with chief cells, because I'll confess I did that in the past, and it was a mess. Here's what you got to look for. If you see here, all these follicular cells, okay, are stained dark. This cell is not. That's the C cell, otherwise known as clear cell. It doesn't take up the stain, okay? It's usually outside of and sort of in kind of a middle gap between the follicular capsules or cavities, between these little follicles. That's the thing you want to keep in mind. The purpose is always to lower the blood calcium levels, but it does it not only in children, it can do it in adults. Keep in mind that calcitonin inhibits calcium reabsorption in the kidney, therefore that calcium is going to be excreted in the urine. It promotes deposition of calcium into the bones. It inhibits osteoclasts and stimulates osteoblasts. It will lower calcium ion levels in the blood, and it will inhibit calcium absorption by the intestines. Now, some of the cellular effects of calcitonin is that the target organs are both the bone and the kidney primarily. The bone, calcitonin will inhibit bone resorption. It will inhibit osteoclasts' motility and differentiation. It will reduce osteoclasts' acid secretion via altering their hydrogen ATPase activity. Now with kidneys, calcitonin will increase urinary calcium ion excretion by inhibiting renal tubular reabsorption. There are calcitonin receptors. They are mediated through G-coupled protein receptors, and these include the G-alpha S, G-alpha Q, and G-alpha I. Several of the calcitonin receptor subtypes have been identified, and they all bind, interestingly enough, with high affinity for that hormone. The calcitonin binding signals are basically via cyclic AMP and the activation of protein kinase A. So this is what's going on inside the cell. Calcitonin can also activate phospholipase C pathway, resulting in a release of calcium ions from their intracellular stores and an influx in extracellular calcium ions. So, if we take a look at what we saw earlier about the dietary intake of calcium, etc., and we were looking at PTH, then we were looking at vitamin D, now let's look at calcitonin. Now, calcitriol, remember, is another name for active vitamin D. So, you have a diet, you have, let's say, daily a 1,500 milligram intake. The feces are going to pass out 1,300 milligrams. You have the intestine doing some absorption and adding to the calcium pool. Some of that calcium pool may end up being secreted a little bit, but calcitriol, the vitamin D3 or active vitamin D, is playing a controlling role here. But then we have to deal with the calcium pool. Now what we see is this. Calcitonin is going to play a role in the formation or the deposition of calcium into the bone. PTH and calcitriol will be involved with the resorption of bone back into a calcium pool. And then we have PTH, calcitonin, and calcitriol, which play a role in the excretion of calcium. Calcitonin would enhance excretion of calcium in the urine. PTH would do the opposite. Okay? And this is from an older physiology text, but I think it helps very well because when you're trying to achieve homeostasis here, you want to go from 9 to 11 milligrams per 100 milliliters, the deciliter. So what happens is rising calcium levels, you have an imbalance. So you've got the C cells of the thyroid gland producing calcitonin. The calcitonin stimulates calcium deposit in the bones. You have the falling blood calcium levels. And you have an imbalance again. And here's now what you've got. You've got the parathyroid cells making and releasing parathyroid hormone, which will act on the osteoclasts to degrade bone matrix and release calcium into the blood. Now that seems to be pretty straightforward. We've got this sort of compare and contrast little chart here I thought would be helpful for you. But you've got to be able to clarify what's what. What does one do? What do you see when you have normal levels, and what is their response? Where are they located? And the last three help, because what happens is calcitonin indirectly increases the mineralization and synthesis of bone matrix. Parathyroid hormone increases the formation of osteoclasts, increasing bone reabsorption. Then we go to one down below that. Low levels of calcitonin lead to osteopenia. Low levels of parathyroid hormone lead to hyperthyroidism. And finally, what about the opposite? High levels of calcitonin will lead to hypocalcemia. High levels of parathyroid hormone will lead to hyperthyroidism. Now, we're going to look briefly at diseases of the parathyroid hormone production. Hyperparathyroidism is basically just that. excessive PTH production. But there's different types. There's primary and secondary. Primary, this is caused by parathyroid hyperplasia, adenoma, or carcinoma. The clinical signs are elevated PTH levels and increased calcium ion levels. This is where you're going to get hypercalcemia, as well as increased urinary calcium excretion, and that's hypercalciuria. Now, one thing to keep in mind, you're going to have the increased formation of kidney stones, that's the urolithalysis, as well as decreased phosphate levels. When you have more and more calcium showing up in the urine, that increases the risk that you're going to start having kidney stones. Now, for hyperparathyroidism, the secondary, this results from alterations outside of the parathyroid gland. It could be a complication in patients with chronic renal failure. Usually what happens is sort of the early stage, you have a reduction of plasma vitamin D and decreases in ionized calcium levels. These are going to contribute to greater PTH levels. Later on, in the further stages, PTH expression of vitamin D and calcium ion receptors is reduced. This results in the reduction of the negative feedback regulation of PTH release. So you move from having some control to losing it as everything is beginning to get losing its influence. There are a couple of other brief charts here explaining or clarifying primary hyperparathyroidism. Usually what you're looking at for primary hyperthyroidism, it might be the hyperparathyroidism. You'll have adenomas or hyperplasia. Secondary, it is a physiological compensatory hypertrophy of all parathyroids because of hypocalcemia. In other words, as the calcium levels are dropping for other reasons, such as chronic kidney disease, vitamin D deficiency, etc., the PTH levels are going to be raised. Unfortunately, the calcium levels are low or normal. The causes of primary hyperparathyroidism, 85% is caused by adenomas. 15% is parathyroid hyperplasia caused by, of course, the hyperplasia of that area. And then finally you get maybe about 1%, extremely rare cases of parathyroid carcinoma. Now, here are some symptoms that you would see. Be aware of them, because you also have case studies this week. With the building up of calcium levels, you're going to have in the nervous system fatigue, depression, confusion. In the urinary system, as I said before, you're going to have kidney stones, an increase in thirst, and an increase in urination. Digestive system will be loss of appetite, nausea, vomiting, constipation. Musculoskeletal system, the muscles will demonstrate weakness, and there will be aches and pains in the bones and joints. Now, what about the opposite? Instead of hyper, we deal with hypo. Hypoparathyroidism, this is when you have low or no PTH production. This can be the result of a surgical removal of the parathyroid glands. It can be associated with other endocrine disorders and neoplasms. It can result in hypocalcemia. Now, the early manifestation of the loss of PTH has also been found to be hypocalcemic tetany. Tetany is just this extremely hypersensitivity of the nerves. One classic clinical symptom is called Chauvet's sign. Basically, it's a twitching or contraction of the facial muscles in response to just tapping the facial nerve at the point anterior to the ear and above the zygomatic bone. Now, there are several signs. They are mentioned, and I actually try to include some videos to give you some demonstrations, such as Trousseau's sign and Chauvet's sign. When we talk about hypoparathyroidism, as the levels of PTH drop, the levels of calcium drop. Now, what could cause this? There's a bunch of different ones. Two right off the bat is if you try to do parathyroid surgery or thyroid surgery. Early in surgical success, when a patient had, let's say, a thyroid tumor, they would tend to remove the whole thyroid. When they had to, they tended to also get the parathyroid glands as well. Then the patient, after the end of surgery, would start having these wildly swinging ranges of calcium levels in their blood. There are some causes that are autoimmune, familial, idiopathic, and infiltrative. Main points to keep in mind when you deal with hypocalcemia are the tetany, Chauvet's sign, Trousseau's sign, the paresthesias, and this is where they're talking about pins and needles on their fingertips, strange feelings, etc., toes, feet, hands, and also some cardiac irregularities, a prolonged QT interval. With hypoparathyroidism, it commonly will occur accidentally after surgical removal of the thyroid gland. It may be latent or overt tetany, also characterized by hypersensitivity, low threshold of the nerves and muscles. And as I mentioned to you before, can be demonstrated by two signs. Chauvet's sign, tapping on that facial nerve as it emerges from the parotid gland in front of the ear. You're going to get a contraction of the facial muscles on that side. Trousseau's sign, usually what you're going to have is someone that might be using a blood pressure cuff. So they arrest or block the blood flow to the forearm for a few minutes. You'll see this flexation of the wrist, thumb, and metacarpal phalangeal joints. Now, this gives you sort of a greater rundown of some of the symptoms you're going to see in the various parts of the body due to hypoparathyroidism. Keep in mind several things. One, some of them will show off much earlier, whereas others would show off much later after the damage to the parathyroid glands. So things like dry skin and coarse thin hair and pustular psoriasis, kidney stones, nephrocalcinosis, chronic kidney disease. A lot of these might take some time. Cardiac arrhythmias, seizures, calcification, Parkinsonism, dystonia. It depends on whether the patient is being monitored post a thyroid or parathyroid gland removal. Okay? All right, as always, we're going to have case studies this week. So what I'm going to do is ask you, because the file was produced a little later this week than I'd like, what I would ask you to do is upload the completed case study by Friday by 4:59 p.m. Okay? I'll move the date back a little bit. We will meet still on Thursday, all right, at 5 p.m. East Coast time. In all cases, please try to work on being complete, accurate, and correct. Okay? And remember that these sheets will be helpful for you to prepare for the next exam. Okay? And finally, next week we will cover the adrenal gland. Have a great day.